Hello, everyone. My name is Hector CARCAMO. I am a research scientist at the Lethbridge Research and Development Center. First of all, I would like to thank all the organizers for the privilege to be here with you and be able to tell you a little bit about the work that we're doing in Canada on the topic of biological control of insect pests in canola. So the title of my presentation is a little bit different from what is listed in the program. I'm going to be telling you about cabbage, seed pod weevil, and lygus bugs, and not so much about coleopteran insects in general, because we really only have the cabbage seed pod weevil as the main pest in southern prairies. We do have lots of issues with flea beetles, but flea beetles are very difficult to control with biological control. So my talk will be about seed pod weevils and lygus bugs. And first of all, I would like to show you a slide showing the collaborators for this project. This is a fairly large team. It's a collaboration between scientists from Agriculture and Agri-Food Canada and academia. I am in Lethbridge in southern Alberta, as you can see here. And also in the city, we have Dr. Dan Johnson, and we have a graduate student working on landscape ecology of cabbage seed pod weevils. Her name is Piratipa Jigatuswaran. Then if we move a little bit east, we go to Saskatchewan, where we have Dr. Megan Vankowski and her technician Jonathan Williams. They're also with Agriculture and Agri-Food Canada. And then in the east, we have a fairly large team of colleagues working there. With Agriculture and Agri-Food Canada, we have Dr. Pat Bouchard, Dr. Tara Gaviepi, and Dr. Peter Mason. Some of them are in Ottawa, others are in London. And then the farthest east, we're working with staff at Université de Québec à Montréal. There we're working with Dr. Geneviève Lagorie, Dr. Eric Lucas, and they have two graduate students, Marie D'Ottavio, doing a PhD, and Claudine Desroches. So that is the very large team, and a lot of the data that I will show you here is unpublished data, so please do not reproduce the slides. So next I want to show you the short program for my talk. I will give you a little bit of background on canola production, and then I will discuss the two case studies on current classical biological control with cabbage seed pod weevils, and then the lygus bugs, which is more of a neoclassical biological control approach. Then I will hope to answer this question, to relocate or not to relocate. I will not have definitive answers, but I hope to give you some food for thought in terms of the environmental and economic considerations. So let me show you a little bit about the canola production in Canada. We have something like 32 million hectares in Canada, and 85% of that is arable land that we have in the prairies ecozone. And as you can see from this slide, canola is a very important crop. In 2017, for example, we had something like 42% of the agricultural area was canola. So canola is a very important crop, and we have a couple of important insect pests that we have to contend with. We have the cabbage seed pod weevil and the lygus bug. Depending on when you seed your canola, you could run into issues with cabbage seed pod weevils. For example, if you plant early, you will have cabbage seed pod weevils that you probably will have to control. If you plant late, you will have lygus bugs that will become a problem. So let's talk a little bit about cabbage seed pod weevils. This weevil arrives in canola crops when the crop is at the bud stage, and they will peak around the early flower stage. That's when we recommend monitoring and controlling this pest. The main economic problem from this weevil comes from the damage that the weevil causes to the seeds. They eat about 3 to 5 seeds, which is about 20% of the crop is consumed by this pest. And so far we have been relying on chemical control. However, we have learned that in Eastern Canada, this is work by Dr. Geneviève Labrie on published data. She has monitored fields of canola in Quebec, and they have found that the parasitism level can be quite high in some fields. In fact, about half of the fields had parasitism by Trichomalus perfectus. This is an adventive parasitoid. It arrived in the area without anybody actually introducing it. It's a welcome accidental visitor, I guess. And in some fields, we can have up to 100% of the larval parasite. So this is very, very attractive. So we are interested in studying this in more detail. And hence we have this goal of conducting a complete economic and environmental assessment of the situation in Quebec. And then you would provide data to support or reject a petition to relocate Trichomalus perfectus to the prairies. And some of the specific objectives include, as I mentioned, doing an economic and environmental assessment. And also we would like to identify which ones are the potential non-target weevils and parasitoids that we have in Eastern Canada and also in Western Canada. In addition, we are working on biogeographic factors. We are refining a Climex model, and also we are considering the landscape attributes that may affect the distribution of these insects. So now I'm going to focus mostly on the potential non-target weevils and parasitoids, and I'll show you some of the work we're doing. Our colleagues in Saskatchewan have been busy in 2021. Here's a map showing the many of the sites that I have been visiting. And here we have a very nice long list of the plants that Dr. Dan Johnson has been confining in various containers to rear weevils and parasitoids. And they have been doing this work also in Quebec. So now I'd like to show you kind of a summary list of the weevils that we have found so far, or our colleagues have found. And here you can see, of course, in all three areas, Quebec, Ontario, and Alberta, they have found Pseudoryncus obstrictus, of course, is the pest. And they have found very few weevils in general that are present in either in canola or in non-cultivated Brassicaceae. There's a couple of weevils that are native to Canada, Pseudoryncus neglectus, and this was in fact listed as a potential minor pest of canola, because this native weevil actually does invade canola fields, and Lloyd Dostal in 2012 listed it as a potential pest of canola. The other one that perhaps is more of a conservation concern is Pseudoryncus omnissus. This species has been found in Ontario and Quebec, and it seems to be heavily parasitized by Trichomalus perfectus. It is listed as a species that should occur in the prairies, but in the rearing that we have done so far, our colleagues have not found it yet. So it seems to be a fairly rare species. There are other species, Pseudoryncus sericimi. This is an exotic weevil and shepherd's horse. And there's also, I believe, Pseudoryncus typhae, which occurs in the east, but does not occur in the west. And this is an exotic species that could be a potential biocontrol for shepherd's horse. So those are the weevils that we have found. Let's take a look at the parasitoids. And for the parasitoids, special thanks to Claudine Desroches and the rest of the team in Quebec for sharing this data with us. Claudine sent us the data. They had 274 boxes that they used to rear parasitoids and weevils. And out of the 110 parasitoid wasps that emerged, only 2 of them were Trichomalus perfectus. One of them came out of Pseudoryncus neglectus from wild mustard. The other one came out of Pseudoryncus typhae, which, as I mentioned, it feeds on the seeds of shepherd's horse. So with this information, we could start making some preliminary conclusions to attempt to answer this question: to relocate or not to relocate. So we know from work that has been done by Tim Haye and colleagues in Europe that the parasitoid is oligophagous, but mainly feeds on seeds, on larvae that feeds on seeds of Brassicaceae, and it's not really found in other plants that are not Brassicaceae. So we would expect negligible effects on non-targets, and it seems that there are not that many weevils, native or exotic, in the prairies that are on seeds or even stems of Brassicaceae plants. A very important component in this kind of analysis is to do a cost-benefit analysis. What is the cost of doing nothing? You know, if we continue the widespread use of insecticides, we know that there are environmental and economic costs. Right? There are effects of insecticides that are well known on pollinators and natural enemies. So that's something that needs to be taken into consideration. So once we complete the current studies, our team will likely submit a petition for relocation. So that's the case of the KBC for weevil. Now I'd like to take a few minutes and talk about lygus bugs. Lygus bugs are a native complex of insects. There's different species depending on where you are. They're very polyphagous. They will go after canola, seed alfalfa, many crops. They're a pest of cotton, strawberries. Almost hard to find something they will not feed on. So they're very polyphagous. So lygus bugs in the prairies, in canola, we often rely on aerial insecticide spraying. Once you have 2 or 3 lygus bugs per strip at the early fall stage, usually people will control them with insecticide. So we're very interested in biological control of this insect also. And we have been following very carefully and lots of interest what's happening in the Northeast USA, where they released Pristinus digonudus. This parasitoid came from, I believe, from France, and excuse me, it has done very well in the Northeast US, where they have noticed quite a dramatic decline in the number of lygus bugs in their alfalfa, and it also has moved to other crops, and it even moved into Canada. It didn't bother to get a passport, it just crossed the border, and it's in Quebec, and it's very well established there, and also in Southern Ontario. So the question that we have is, should we bring this parasitoid to the West? Is it going to establish? Is it going to cause potential issues with conservation of native parasitoids? So this is something that we're very interested in pursuing, and we have been working on this for a while. So I'd like to just share a little bit of what we have been doing and plan to do. Now, this graph has quite a bit of information, so let me spend a couple minutes describing the graph. Here we have a generalized relative abundance of lygus nymphs, and the parasitoids attack the second and early third instar. And we do have native parasitoids in the prairies. We have at least 3 species that are found on lygus bugs. The first generation of lygus nymphs available for the parasitoids will occur in forage crops like alfalfa, and also in weedy areas, non-cultivated areas. And this generation is not as large as the second, but this is the generation that gets parasitized most heavily by Peristinus dei and Peristinus melipes, native species. But the timing of the attack is very narrow. So you will find a very specific cohort, so that if you take a look at the entire population of lygus bugs, you will only find around 5 to 10%, maybe 10% of the entire population will be attacked by Peristinus wasps. So if we move on to the second generation of lygus that occurs in the southern prairies, sometimes you may even have 3 like this year, but usually you have 2 generations. This second generation occurs mostly in canola, and there's still some that occur in perennial forage crops and other uncultivated areas. Now, this generation is huge because lygus tend to explode in these other crops, especially if the weather conditions are suitable for the development. And here, interestingly, we have very low levels of parasitism. There's only 1 species that attacks this second generation, Peristinus howardi. And in canola, we find pretty much 0 parasitism. In alfalfa, we can find perhaps 5% parasitism, so very low levels. So our question is, what happens if you bring Pristinus digonudus? We know that this species, digonuda, has 2 generations, so there is a potential for them to compete with the native species that attack the first generation. And we know that digonudus has a second generation, so that's a bonus. They will be there to attack this larger generation of lygus, and some data from Ontario suggests that they do get into canola, but this is something that we like to pursue and confirm. So this makes it interesting, and it looks like there is a huge niche, a very large population of unexploited lygus nymphs available in many of the crops because they're so generalist. So we have a hypothesis that we're testing, and the hypothesis is that we like to test and see what kind of competition may happen between Peristinus digonudus and Peristinus melipes. Some of this work has been done in Ontario by Simon Lachance, and their work, they work mostly with Geoffran and other native species, and it looks like there might not be as much competition there from the laboratory studies. The field data, I think, might be suggesting otherwise, that the dominant species now is Ligonudus, which I guess is not unexpected. In our case, we are doing laboratory trials. We are looking at what happens if you introduce the two species simultaneously, and we're also looking at sequential competition assays. And we have been trying to do this for a couple of years. Because of COVID, we got delayed a little bit, and getting this data has been rather slow and rather challenging, because working with the native species and rearing them is a little bit difficult, but we are making some progress, and we can share a little bit of results that we are seeing, and the results so far are encouraging. We are seeing that in competition studies, for example, let me go back. For example, if we have a sequential study where we present nymphs to the Peristinus Ligonudus first, and then to the native species, or vice versa, we are seeing that some of the adults of the native species are emerging despite having the nymphs attacked by the exotic species first. So next year, we hope to continue these studies. And here is a bit of a preliminary conclusion and sharing some of our future work ideas. So far, the data suggests that there isn't really a competitive disadvantage for the native Peristinus mellifes, and we are planning to extend the study and apply it to the other native species. And we also would like to make these studies a bit more complex by including Adelphocoris bugs, which are preferred by some of the early native species and not by the Peristinus ligonudus. So that could be a refuge for the native species also. So in the future, we're planning to continue our competition tests. We plan to expand our survey of the native Peristinus wasp. We would like to see what's happening in the new emerging crops. We are seeing other crops like hemp and quinoa are appearing in the horizon here. So we'd like to see if the native species are there, and also what is the LIGO situation there. And once we complete all this work, similar to the work with the Cabecito weevil, we're planning to write a petition and see if we can relocate this native, these exotic parasitoids that are already in the east in the country and move them to the west. So hopefully they can help us to manage these insect pests in a more sustainable way. And with that, I'm going to thank you all for visiting and listening to this presentation. And I also like to acknowledge the financial support we get from the Canola Council and the Canola Producer Commissions throughout the Prairie provinces. And especially I'd like to say a big thank you to all our graduate students, technicians, summer students and interns that help us. Without their help, we could not have the data and the information for me to give this presentation. Thank you.